Okay, so where we're going to start today is with the fermentation and anaerobic respiration. So what we were looking at last time was your aerobic respiration, meaning it's done in the presence of oxygen. Even though glycolysis does not require oxygen, glycolysis is required in order to be able to do the citric acid cycle, which does require oxygen. So things are usually either going to take that pathway where they're going to go with glycolysis and into the citric acid cycle in the electron transport chain, or if oxygen is not present, they will go with a different cycle with the fermentation cycle in there. So certain anaerobic prokaryotes are going to have electron transport chains that do not use oxygen as a final electron acceptor. They're going to be slightly different. We're just going to acknowledge they exist because they're not really going to be too important for you in there. There are microbes that will consume all kinds of substances, but one example would be the sulfate-reducing bacteria, that they're going to use hydrogen sulfide instead of, or they'll produce hydrogen sulfide instead of water at the end, so it's H2S. It replaces sulfur with the oxygen in there, and being able to process that. So those are important for the environment, but in terms of going into healthcare right now, other than they exist on our planet that we need to stay alive on, we don't really use them for anything. So what fermentation is going to do is it's going to harvest the chemical energy without using oxygen or an electron transport chain. What it does need is a sufficient supply of the NAD plus to accept the electrons during glycolysis. So when we look at fermentation, you want to keep track of the electrons in there, that's more where your focus is rather than looking at what is happening to all of your carbons and the phosphates in there. So types of fermentation, alcohol fermentation in there. Here you take the pyruvate that you got from glycolysis, and it's going to release carbon dioxide to make a acetaldehyde, and then that gets reduced by NADH to ethanol, and you're going to have to regenerate the supply of NAD, is what this should say. NAD plus to continue glycolysis. So you can, if you remember back in glycolysis, there was an NADH that came off of there. In order to keep that going in there, you have to have a supply of NAD plus that gets turned into NADH. And you have plenty of that going on with the electron transport chain, but when you do fermentation, the end stage of fermentation in there is going to provide more of that NAD plus to keep the cycle going in there. So alcohol is one thing that is fermented. Beer and wine are fermented for the alcohol in there, and these are the types of reactions that go on to do that. In the human body, we will have lactic acid fermentation in there, and you'll take the pyruvate that is going to be reduced directly by NADH to make lactate. It doesn't release any carbon dioxide when it does that. So this is what you're going to do if oxygen is not present to supply your muscles. You'll get as far as glycolysis making the pyruvate, and you can't continue into the citric acid cycle. So you have to go with plan B, and that's going to be the lactic acid fermentation in there. So lactate can be converted back to pyruvate by your liver cells in there. So that's how your body is going to eliminate lactic acid in your body. When you see lactic acid and lactate, think of them as the same term in there. They're both acids in there. Chemistry likes to have lots of names for things too. So it used to be thought that the lactic acid was the primary thing responsible for your delayed onset muscle soreness, why you would have the soreness in your muscles a couple days after exercising. New research is really showing other things are more to blame for that. Your increase in potassium levels may play a role in there. We haven't wrote off lactic acid playing a role in there at all, but it does not play the primary role that they've thought for a long time in there. So when we compare aerobic respiration and fermentation, both of them are going to use glycolysis to oxidize the glucose to get to ATP. So they're both going to start out with that beginning part. NAD plus is going to be the oxidizing agent that accepts electrons from the food during glycolysis. So both of them use that NAD plus. How they're different is in how they oxidize NADH back to NAD plus to sustain glycolysis. In fermentation, you're going to have your final electron acceptor be an organic molecule in there, pyruvate for lactic acid, acetaldehyde for alcohol in there. With aerobic respiration, the final electron acceptor is oxygen, and you get a lot more ATP out of that. If there was a part of this that I was going to say you will hear over and over again, the key part is what is the final electron acceptor that they look at in here. That if in all of this, this is probably 90% of the time which you would be asked about fermentation, comparing it to oxidative metabolism, is that difference in there. Aerobic respiration is a lot more efficient. It's going to give you 19 times more ATP than fermentation does. So anytime an organism can do oxidative metabolism, it's going to choose to do that over fermentation. So when we look at the oxygen requirements, we can put organisms in different categories. An obligate anaerobe is going to carry out fermentation or anaerobic respiration, and they cannot survive in oxygen. So obligate means that it's a required in there. They have to be in anaerobic conditions. Part of the reason they have to be in the anaerobic conditions is the oxygen is toxic and destroys them. And they're going to do fermentation for metabolism in there. Obligate aerobes have to have oxygen in there. They only carry out aerobic respiration and not fermentation. As humans, we do some fermentation, but the reason that we're obligate aerobes is the brain is not capable of doing fermentation. So you have to have oxygen to keep your brain alive. Your muscles could actually function not very well doing glycolysis without oxygen and fermentation for the lactic acid. They're not going to produce much energy, but your brain absolutely has to have the oxygen in there. Facultative anaerobes, these make enough ATP to survive using either fermentation or respiration. So they can go either way, and our muscles actually fall into that category. So the evolutionary significance of glycolysis. It's thought that the ancient prokaryotes probably used glycolysis to make ATP before oxygen was present in the atmosphere. And then the cyanobacteria developed, which produced oxygen, and it's thought that's how the oxygen got into the atmosphere, is a byproduct of them being able to do photosynthesis. And then that opened the door for aerobic metabolism to come in. So we obviously do consume things other than just carbohydrates in there, and ones other than glucose. Glycolysis is going to accept a wide range of carbohydrates. So if you are a sugar other than glucose, what's going to happen is you're going to get hydrolyzed to glucose or other intermediates in the cycle and join in. So you've got that 10 steps in there. The other carbohydrates are going to join somewhere in along those 10 steps. Proteins, on the other hand, they have to be broken down into their individual amino acids. Most of your protein actually is going to be used to build other structures. You can use it to derive a little bit of energy, but your body will choose to use it for structures first. And it will choose to use it for energy last thing in there. So if you have excess amino acids, your body's going to try and say, well, can we use any of this for energy? And some of them will be converted into the intermediates of glycolysis. Some of them are going to be converted into intermediates of the citric acid cycle. The ones that end up being intermediates of glycolysis, you can actually convert back into sugar and fuel the brain with those a little bit. But anytime you break down a protein in your body, which is going to come from muscle and liver, you're going to get a mix of them. The other ones end up having to go in through the citric acid cycle, and they produce ketones as a byproduct that is going to give a person that distinct smell. If you've been in a situation where you've had to sustain activity for several hours and not been able to have adequate food, and you may notice that you don't just smell the normal sweaty. There is a different distinct smell to sustaining long activity like that. And what you're smelling are the ketones coming out in your system. So when you break down amino acids, you've got the amine waste on there that you have to deal with. So the Krebs cycle or citric acid cycle, it's going to be looking at breaking down carbons in there. It has no means of dealing with nitrogen waste. And so that nitrogen waste has to go through deamination, which just means we pull the amine group off. And it gets converted into urea that is going to eventually be excreted as urine from the body. Triglycerides. So if you remember, a triglyceride is a glycerol with 3 fatty acids attached to it. The glycerol only has 3 carbons on it, so that's going to go into glycolysis. It gets converted into the glyceraldehyde 3-phosphate. So you can use a little bit of the glycerol portion to fuel your brain if you need to. The fatty acids, though, which are the majority of it, end up getting split off doing beta oxidation. They get pulled off 2 at a time, is what beta oxidation means. And they're converted into acetyl-CoA, and that's where they're going to enter into the citric acid cycle. So when you look at it, a typical fatty acid that a human would consume is going to have between 16 and 24 carbons on there. So if we say we've got 16 carbons on there and we're doing beta oxidation, how many acetyl-CoAs are you going to get? Pulling them off 2 at a time. So that's going to leave you 8 acetyl-CoAs. And a triglyceride means you've got 3 of those fatty acids in there. So 8 times 3, now we're up to 24 acetyl-CoAs. So if you consume a glucose, you have to go 2 turns of the cycle to burn that up. If you consume a triglyceride, it's going to be 24 turns of the cycle. That's why fats have so many more calories in them than carbohydrates do, is because there is more carbons that you have to break off in that cycle. So biosynthesis, this is anabolism or building things. So once we break all this stuff down, your cells are going to use some of the carbon skeletons to make their own molecules. So some of the carbons that you consume in there are not actually going to end up going through the Krebs cycle. If you need it as a building block to build something else in your body, it will just take it. With the amino acids, if your body needs to make a new protein, it's just going to take the amino acid and use it to do that. Then it doesn't have to go into that cycle and be completely broken down. The ideal thing is to try and figure out how much protein it takes for you to be able to sustain repairing your body in there and consume that amount of protein, because anything else is going to put more strain on the liver and kidneys to break it down. Your intermediates of the citric acid cycle in glycolysis, a lot of times before they finish the cycle, they can be diverted off and be used to make other things when the cell needs them. And anything excess that you eat, carbohydrates, proteins, fats, it's all converted to fat stores. A lot of times people are under the misconception that well, I'll just consume extra protein and it's going to turn into muscle. Your body is not going to make muscles that it doesn't feel you're going to use. It will just turn it into fat. If you consume extra fat that you don't need, it's going to turn that into fat. If you consume extra carbohydrate you don't need, it turns that into fat. All pathways lead to making fat with too much. And there's no way around it, unfortunately. So most of what we do in metabolism is going to be regulated through feedback mechanisms. Most is going to be feedback inhibition, where at the end of that anabolic pathway, if you build up enough of that product that you don't need it anymore, you're going to use an enzyme to inhibit something earlier in the pathway. So you go back to the beginning and say, okay, stop here and there. Catabolism is largely regulated by negative feedback levels with ATP. And so when you start to have enough ATP in the system, what's going to happen is it will shut something down earlier. And there's a few examples of that. You can have it shut down phosphofructokinase in step 3. That enzyme is going to be inhibited by ATP. And then when you start to deplete the ATP to ADP and then AMP, it will stimulate it again. It's going to say we're running low on ATP. Let's speed up metabolism a little bit in there. Phosphofructokinase is also sensitive to citrate in the citric acid cycle. And that helps keep those two cycles coordinated in the speed that you're going. If you have the citric acid cycle going too fast, a lot faster than glycolysis is, you're not going to have anything to feed into it. And likewise, if you have glycolysis going too fast, you're going to start to build up all of this pyruvate in there that you end up having to turn to lactic acid instead of letting the citric acid cycle use it. So with photosynthesis, we'll get started on this today. I did put up some pictures for this last night that might be helpful to you with that. In the biosphere, which is the whole planet where you've got living life, all places that have the living life, photosynthesis is going to be the process of using light to make chemical energy. And you store that chemical energy in sugars and other organic molecules. Autotrophs are considered to be self-feeders. They are able to produce their own organic molecules from carbon dioxide. So plants are considered to be self-feeders in that they can make organic molecules from the carbon dioxide in the air in there. So they can use other inorganic raw materials from the environment, but they don't need other living beings to do that. So they'll use minerals out of the soil, but they don't need other living beings to do that. And that is a real simplification of things, because plants obviously need something more that I don't give them to survive in my house, but there are some plants that are going to be dependent on bacteria to live as well. The cycle is going to need other people to regenerate the carbon dioxide, to regenerate the minerals. But what they actually use right then, directly, immediately, is going to be inorganic materials, and they feed themselves by making their own sugars in there. So heterotrophs, hetero means different, variety of things. These are going to obtain their organic materials by consuming compounds produced by other organisms. So in the scheme of things, they're considered the consumers, where plants are the producers in there. We are heterotrophs in there that are going to be consumers. So the photoautotrophs, these are going to use light energy to derive the synthesis of organic molecules from carbon dioxide. Those are your plants in there. That's where they get the energy from. So when we look at plant cells, they've got chloroplasts, which is going to be the site of photosynthesis in plants. So what organelle in animals do you think that would be similar to If that's going to be where they're making their energy, what did animal cells use to make their energy? The mitochondria. And when you look, the chloroplasts actually have some DNA in it, just like the mitochondria do. Chlorophyll is going to be the tissue in the interior part of the leaf where the chloroplasts are found in there. And the mesophyll is going to be that middle part of the leaf. It's going to have microscopic pores where carbon dioxide is going to enter the leaf and oxygen exits. The roots are going to absorb water and some of the minerals in there, and it's going to deliver it out to the leaves through the veins. So it's only the green part of the plant that's going to be doing the photosynthesis in there. In the leaf, we've got these thylakoids, which are systems of interconnected membranous sacs that are going to segregate the stroma and chlorophyll in the thylakoid membranes. So the grana or granum are stacked columns of thylakoids found in some places. In a lot of pictures of them, they kind of look like stacks of pancakes that are in there. Just these little stacks of chlorophyll in there is how they draw them. So if we look at what happens to the atoms going through photosynthesis, you start out with 6 carbon dioxides, 12 waters, and light energy, and you make glucose, 6 oxygens, 6 waters. And when you look, it's almost the exact opposite in there of what aerobic metabolism does. We break down sugar and get these things out of there in energy. So glucose is used to simplify the equation, but if you manipulate it through chemistry, it actually produces a 3-carbon sugar in there, but when we're looking at it, we talk about it as a 6-carbon sugar so that the numbers compare well to the 6-carbon sugars that we use with glucose. So this is simplified even more in here. You start with carbon dioxide plus water. And sugars, when you have 6 repeats of this pattern in there to make a sugar as a way of manipulating it in there. If manipulating that part of the chemistry doesn't make sense to you, this works just fine to understand that you use carbon dioxide and water with light energy to make sugar. So the process starts out with splitting water. And so oxygen is going to be given off from the plants by the chloroplast splitting water into hydrogen and oxygen. That's how we get the oxygen in the air. When they looked at this, C.B. Van Niel proposed that all synthetic organisms require hydrogen, but they've actually found that they don't, that you can have the sulfur bacteria out there will use sulfur in there instead to produce things. So they've changed it more to a general reaction that you have carbon dioxide plus hydrogen oxide, which is water. You can have hydrogen sulfide here. Hydrogen blank in the space can also be used. So that general reaction fits for lots of different organisms. What we are going to be spending our energy looking at is this one here for the plants. It is a redox process in there, which means you're going to be moving electrons back and forth in there. In respiration, energy is released from the sugar when the electrons associated with the hydrogen are transported by carriers to the oxygen. In photosynthesis, your electrons are going to increase the potential energy as they move from water to sugar. So it's kind of like you take the reverse of the electron transport chain with the sunlight in there to harness the energy. The electron transport chain, we're taking the energy out of it. What sunlight is going to be done is it has to harness the energy in the reverse direction of what's going on. So in photosynthesis, we have two stages. The first stage that goes on is the light reactions in here. This is going to be where you take the energy from the sun and you convert it into chemical energy. So similar to NAD, they use NADP+, nicotinamide adenine dinucleotide phosphate. Calling it NAD+ is the way you'll see it all the time. That's going to be your electron acceptor, just like you had NAD+ as an electron acceptor. So what solar power is going to do is reduce the NAD+ to NADPH in there. We're going to have photophosphorylation, so we're going to use those light reactions to generate ATP. And it's going to be done through chemiosmosis in there as well to provide the energy for the ATP. The second part of the cycle, you're going to have the Calvin cycle in there. It's sometimes called the Calvin-Benson cycle. It's going to take carbon dioxide from the air, and it's going to basically glue them together to make sugars. So it converts it into organic molecules in the chloroplasts, and then those get reduced to being carbohydrates by adding electrons on there. And you're going to get the electrons from NADPH in there. So solar energy to charge up these NADPHs with electrons. Then you're going to use those electrons to provide the energy to glue these carbons together at the end to make the sugar. So when we talk about carbon fixation in there, it's the initial incorporation of carbon into the organic compounds. So you can think of this like where you add the acetyl-CoA into the citric acid cycle. You're adding the carbons into the cycle, and then you've got to split them off later on. When we fix carbons, here we're adding carbon dioxide into this cycle to make sugars. And then we say the carbon is fixed when we put it into that cycle. So when we look at sunlight, it is light, and one of the properties that light has is it's going to be in waves. And the wavelength is going to be the distance between the crests of the waves. So when we talk about the size of waves, one of the ways we measure them is their amplitude, how tall they are, but how far it is between the tops of the waves. And the distance between the tops is the wavelength in there. We've got the electromagnetic spectrum, which is a range of wavelengths. So you've got really small ones that are just in nanometers that are going to be your gamma rays. And then you've got really big waves at the other end that are more than a kilometer. Those are going to be your radio waves in there. And visible light is a very small section of it that is going to range from about 380 nanometers to 750 nanometers. And one of the things we know about visible light is it not only behaves as a wave, but it can behave as a particle. And when we're talking about it behaving as a particle, we talk about it as a photon. So photons are your discrete particles of light. They're not tangible, but they do have a fixed quantity of energy. The reason light gets considered to be that is you've got the theory of relativity where energy and mass are converted. So if you've got energy, it has to behave like a particle in there because it's a different form of mass in there. So the amount of energy is inversely related to the wavelength of light. The shorter the wavelength, the greater the energy of each photon in there. And so we have different pigments in there. When you're looking at a color, what you're seeing is the light that is reflected from the pigment. And so part of it is also going to absorb the visible light in there. Different pigments absorb different wavelengths. and the absorbed wavelength disappears in there. So when we look at green leaves, what we're seeing is the green being reflected. The other things around it disappear in there. You can use a spectrophotometer to measure the ability of a pigment to absorb various wavelengths of light. And you actually can plot out these absorption spectrums. And there's 3 pigments that we have in the chloroplasts. So chlorophyll A, chlorophyll B, and the carotenoids. Chlorophyll A is going to participate directly in the light reactions. Chlorophyll B is an accessory pigment. It helps to extend the wavelength of light that you can absorb. So by having chlorophyll B in there, you're able to absorb more energy from the sun rather than just in a narrow wavelength in there. And then the carotenoids are also groups of accessory pigments. They tend to be orangish red in color. So one of the things when you see leaves that die, a lot of times you look at that and you go, well, they change colors because they're dead. What happens is the chlorophyll A and B are going to dry up in there and no longer be visible. And what you're seeing with those red colors in the leaves then is actually the carotenoids in there that are available in much smaller amounts, but when you take the other things out, now you can actually see the carotenoids in the leaf. Is what's left behind. And so when we look at the pigments, we'll say it has an action spectrum in there, a place where it's going to be most effective at absorbing a wavelength of light. So for chlorophyll A, it's a violet, blue, and red light that works best. Chlorophyll A alone is what they use to measure a lot of things, but it really underestimates the effectiveness because you've got the accessory pigments in there. So it's more than just red and blue light that will help with the plants. That's going to be a good stopping point for today.